the research office for inviting me here, and also all of you for coming. When I was putting this slide together, or this program together, I actually realized I had never given a talk about my research at Montana Tech, except when I interviewed here. And so I'm assuming this will be less stressful, hopefully. So I always like to start out with a little overview of what I'm going to talk about, so that way halfway through my talk you won't go, where the hell is she going with all of this? So you'll kind of have a feel. But I'm going to start out with some background information on hantaviruses. Stella said the hantavirus, and hopefully she will learn that she is wrong in saying that. So I'll give you a little background information on hantaviruses, what they are, why we're interested in them, why you would want to study them. Then I'll talk about some disease ecology research that I've been involved in. I've done a bunch of different stuff. I've worked on longitudinal studies in what we call sylvan, even though that means wooded. Generally what we're referring to is more natural or wild habitats. And then since coming to Montana, I've done a lot of work in peridomestic settings, which are populations of mice that live in and around human dwellings, buildings, outbuildings. And then if I have time, hopefully I'll talk a little bit about some recent work and then where I'm taking this in the future. So I sometimes talk too fast. I drink a lot of coffee, so throw something at me if I need to slow down. But basically, what exactly is a hantavirus? Hantaviruses are actually a whole genera of virus in the family Bunyividae, so they're not just one virus or one thing. Within this family, there are a variety of viruses that cause human illnesses. So there's only one in here, genera, that actually isn't known to cause human diseases. But the top three up here are human diseases that are called vector-borne diseases, which basically means that they are transmitted to humans by insects. And so in the case of, say, La Crosse encephalitis, that's transferred from mosquitoes to people. Crimean Congo hemorrhagic fever is caused by a tick bite. Phlebotomist fever is caused by a sand flea bite. So all of these viruses are transmitted to people by vectors. The other thing I should mention about these viruses, they're all RNA viruses, which just means that for their genetic material, they use RNA rather than DNA. So hantaviruses are actually kind of unique in this family because they don't have arthropod vectors. They actually have rodent host. So that's one of the things that makes them unique. I put a little Trivial Pursuit up here. They're actually named for the Hantan River in South Korea, where they actually were isolated in the 1970s. But in terms of this reservoir host, what we know is there are lots of different strains of hantavirus, and each strain has a different host, and that host is often species specific. So strain A may be carried by this species of rodent. I threw insectivores and bats in here in parentheses because some really recent work has isolated hantavirus, new hantaviruses from shrews, and then also some bat species. Transmission to humans occurs when humans inhale aerosolized virus, and I'll talk a little bit more about that in a second here. And in humans, hantaviruses cause two major illnesses. The first is hemorrhagic fever with renal syndrome, abbreviated there, and the other is Hantavirus Pulmonary Syndrome. So the first one, HFRS, affects the renal system or the kidneys. Hantavirus Pulmonary System affects the lungs. So I mentioned that it is transmitted to people when they breathe in aerosolized virus. So we have chronically infected rodents. Hantaviruses are sort of unique in the fact that rodents are chronically infected, so they never clear the virus. They stay infected for life. They infect other rodents by aggressive behaviors, so biting during reproduction, biting during the reproductive season. So we have mice biting one another and spreading the virus that way. The infected mice then shed the virus in their feces, urine, saliva. People, if that shed virus becomes aerosolized and up into the breathing zone, people can inhale it and become infected that way. There are a couple other ways to become infected: mucous membrane contacts. So if you're handling mice and you rub your eyes or your nose or your mouth, you can become infected that way. Cuts or skin breaches are another mechanism for infection. So in the old world, in Europe and Asia, we actually have 4 hantaviruses that do cause fairly serious human illness. We actually have other hantaviruses in those areas that are not linked to human illnesses. So these are the top 4. These two, where it says severe, moderate, mild, they're talking about the fatality rate when they refer to those. So Hantane and Dubrava virus are severe cases, which means about 5 to 15% of the individuals who get that illness die from those infections. Silvirus is moderate. The fatality rate in silvirus is about 5%, but it's carried by rats. So rats are worldwide. So we do get cases of silvirus in the US, not very common, but it does occur. And then pumalavirus is carried by bank voles. That actually causes a real mild HFRS that has a fatality rate of about 1%. These are all the New World hantaviruses that we have, so there's actually quite a few in both North America and South America. We're going to focus on the US. So in the US, we have a fair number. There's a couple new ones that are not up here on this slide. But prior to 1993, we knew about these two, silvirus carried by rats, and we knew about Prospect Hill virus, which is carried by meadow voles. And Prospect Hill virus is not associated with human illness, so the voles have it, but it doesn't seem to be transmitted to people. In 1993, there was actually an outbreak of a mysterious illness in the Four Corners regions of the desert Southwest, so Arizona, New Mexico, Utah, Colorado. With high fatality rate, it was an illness that affected lungs. People would go into severe respiratory distress, had a high fatality rate. About 50% of the people who were coming down with this illness were dying. So the CDC got concerned. They went in, isolated the virus, and they isolated a new virus, Nombre virus. And then they determined the reservoir host for that virus as being Paradomestic maniculatus, or the deer mouse. Lots of cases, or a fair number of cases of Hantavirus Pulmonary Syndrome since 1993. So this is some summary stats from the CDC. Why the CDC was really interested in this hantavirus, they knew about hantaviruses, was because there wasn't a hantavirus in the US prior to 1993 that caused severe human illness or that caused a lot of human illness. The other reason why they were concerned was this here. So the fatality rate has declined since 1993, and now it's down to about 36%. But that's really, really high for a hantavirus. The highest known fatality rate, as I mentioned in those severe European viruses, was 15%. So now we have a virus that kills about a third or over a third of the people that it infects. So that was cause for concern. The other thing that's sort of interesting about up here, and I'll talk about this a little bit later, is that more males tend to come down with Hantavirus Pulmonary Syndrome compared to females. If you look at this by state, this is pretty up to date. It's been reported in 34 states. What I think is interesting about this is this comparison. So this is the map of deer mice, where deer mice are distributed in North America. So deer mice are habitat generalists. They can live in a lot of different habitats. We find them in a lot of different places. So pretty much everywhere we find deer mice, we have had cases of HPS. So the map is pretty nice here. So most of the HPS cases in the US are caused by Nombre virus. There are a couple other viruses, hantaviruses, that are carried by other species that occur in these southern states, in some of these areas where we do have cases like Florida. So there are cotton rats and rice rats that carry other strains of hantavirus. All right, so we're in Montana, so we want to care more about what's going on in Montana. In Montana, we've had 38 cases with 10 fatalities in 22 counties. And you can kind of see where they are, and we've marked in the fatalities as well. So that's actually a little bit lower fatality rate. The fatality rate in Montana is something like 26%. We'll talk about a little bit, hopefully at the end, if I have some time, to talk about why that might be. So in 1993, following this outbreak, the CDC decided that they really wanted to understand what was going on with that reservoir host in the deer mice. So they actually started funding. I think the funding came through in like 1994 and 1995. They started funding what are called longitudinal studies. And longitudinal studies are done a lot in animals that we know are reservoir hosts, or even in people, they're done. And longitudinal just means that we follow known individuals over time. So we look at when individuals are becoming infected, who is becoming infected, are there seasonal patterns to that? So the states that the CDC decided to fund these studies in were obviously Mexico, New Mexico, and Arizona are obvious choices, and also Colorado. So we're looking at those four corners states where we had that outbreak. For some reason, they didn't fund any in Utah, but Utah had some other studies going on. So in Colorado, they had a couple different studies. Arizona, they had 2 studies, one up in northern Arizona near the Grand Canyon, the other in southern Arizona down by Tucson. That was the study I was involved in. New Mexico had a whole bunch of sites. I think they had 6 or 7 sites. And then Rick Douglas got funding to do work in Montana. So Rick had like 6 different sites that he was looking at deer mice populations in. And so kind of the goal of all this from the CDC's point of view was they really wanted to look at who is infected in these populations. Is it males, females? What are the demographics? Are they adults, subadults? So they wanted to look at the prevalence of infection in different demographic subpopulations of deer mice. They also wanted to look at temporal and spatial differences in prevalence over all of these different sites. And the main reason for that, or the goal of studies that are done this way, is basically to come up with some type of predictive model that lets health officials know when certain areas where mice in certain areas might pose a threat to public health. And so we could have some type of early warning capability to tell people, gosh, you maybe need to watch out for this, tell health professionals that you might have cases of this coming in. Often the goal is also to break that transmission cycle if possible, so to do some type of habitat manipulation or something that can change that cycle or decrease the incidence of human infection. And this picture is actually from the Santa Rita Experimental Range, which is some land that's owned by the University of Arizona, and that is where the study I worked on was conducted. And I think it's really pretty. It does kind of look like Montana, except for the big cactuses and the mesquite. All right, so kind of what the CDC was getting at was something like this. If we know how weather affects vegetation, and we can figure out how vegetation affects insects and rodent populations and how all these things interact in terms of disease transmission, maybe we can predict when human disease will occur. And once we have all this knowledge, maybe we can just remotely census weather and then be able to say, oh, this is bad, we're going to get human disease. Obviously, that's not how it turned out. What we discovered is it's a lot more complicated than that. So there's not an easy way to go from here to here. All right, I'm actually going to go through some of the methods that were used for this study, because it's the same methods that we basically use for all of this other studies that I'll just briefly mention. But if you want to follow known, there's lots of ways to catch mice, right? You can put out the snap traps with a little piece of cheese and catch a mouse, but basically a dead mouse does not, you can't follow a dead mouse over time, right, to see when it becomes infected. And so if you're doing these kind of studies, you have to trap mice, mark them in some way, release them, and then follow them at some point later in time. And so all of these studies use some form of live traps, and some of you in here have seen live traps, some of you haven't, but live traps basically are a little metal box. You put bedding in here, peanut butter, we put peanut butter and oatmeal, different studies use different bait. But there's a little spring on the bottom here, there's a door that flips shut, the mouse cruises in, eats peanut butter and oatmeal, is happy, steps on the little spring on the bottom there, and the door shuts. So it's kind of a nice little home. I've actually lived in worse apartments than these. They're pretty lovely. They're nice and clean. They have food, peanut butter and oatmeal, what's not to like? And then generally most of these studies trap for 3 consecutive nights and then each month. And that's pretty standard for small mammal trapping. Depending on where these studies were conducted, the traps were arranged in different manners. So in Montana, they were set up to use 1 hectare trapping grids. So we have 100 traps spaced 10 meters apart, so 10 rows of 10. In New Mexico and Arizona, we used this crazy trapping web, which covered a lot larger area with a lot harder to trap. So these had something like 500 traps on them. The traps were spaced about the same, about 10 meters, but they were arranged in this spider web or kind of spoke-like pattern. So once we have our mice in the little traps, one of the things we have to do is actually take them out. So generally they're put in a bag. And then depending on where you're working, they're identified to species, and data on sex and reproductive condition is recorded. In the Southwest in Arizona, we caught a lot of different species of rodents. So we would have like 20 different species, so this identification of species sometimes is a little complicated if you're dealing with like 4 species of Paradomestic. In Montana, pretty much all we catch are deer mice. So about 90% of what we capture are deer mice. So that's actually really easy. Mice are generally weighed to give you an idea of the condition they're in. They're marked with these little metal ear tags. So you've all probably seen the cow tags on cattle. They're really similar to that, but obviously a lot smaller, with a lot smaller little numbers. And then a blood sample is taken, and then that's frozen until it can be analyzed, and then the mouse is released where it was caught. And then the blood is actually tested for antibodies. So we're not actually looking for virus in the blood. We are looking for antibody, which tells us that that mouse was exposed to virus. And that's done using a standard ELISA. All right, I'm just going to tell you a little bit. Rick actually, Douglas, talked last year and told you a lot about the longitudinal studies that he did here in Montana. But again, I mentioned these studies were done all over the place, and there were a lot of really good papers that came out of this work. The CDC funded these studies for about 10 years, from 95 to 2005. I listed some of them down here if you're interested. But basically, a lot of the studies, there were some common findings in all of them. And one of these was that we did find higher prevalence of infection in older male mice. So males had a higher rate of infection than females. We also found a positive relationship between wounds and being infected. So that basically is consistent with this idea of horizontal transmission, where we have mice biting one another, passing the virus to one another, especially adult male mice fighting. The other thing that they found is all these studies had different seasonal and multi-year patterns. And one of the things that came out of that was there was a suggestion, and this held pretty well at some of the sites, that there was a delayed density relationship between density of mice and prevalence, which logically makes sense, right? So if you have more mice and mice become infected by fighting, you would think the more mice you would have, the higher your prevalence in that population. That pattern doesn't hold everywhere. In a lot of cases, there is a time lag, so it's not a direct relationship between that. I actually worked in southern Arizona on this, and one of the things that we had there is we didn't actually have a lot of Paradomestic maniculatus. And so our work had some different things that we showed. One of the things was one of the species that had antibodies in our study was a species called the brush mouse. And if I showed you a picture of a brush mouse, it looks just like a deer mouse. They're the same genus, and there are kind of subtle ways to tell them apart. But basically, because of that, the researchers in northern Arizona actually isolated and sequenced a new hantavirus from brush mice. So brush mice don't actually carry Nombre virus, which is the hantavirus we're interested in. They actually carry a different type of virus called limestone canyon virus. The other thing that we found in Arizona is our population of Paradomestic were really low compared to other areas, and so we would get virus appearing and disappearing from the population for really long periods of time. So basically, they would become reintroduced from mice dispersing in from other areas. We would get virus brought back into those populations. But basically, those reintroductions were only successful in maintaining that virus in the population when the density was actually large enough. And Jim Mills, who is a biologist from the CDC, he's since retired, but he's been really supportive of work here in Montana, and then also a lot of these studies. He basically, in 1999, had a summary of all these studies, and in his take on it was, we see all of these differences in terms of spatial and temporal patterns. Really what we need to do is we need a lot more replication of these types of studies. We need to continue long-term studies, and long-term studies are really, really important for this kind of work. All right, so we've been talking about deer mice. What about people? So one of the things that came out of that 1993 outbreak was who is at risk for getting HPS? Obviously males, right? Because males have a higher prevalence of it. But certain people started investigating what are there occupations or situations that put you more at risk for developing HPS? And so they kind of thought about who comes into contact with mice, and they actually tested people for SNV antibodies. So they tested forest workers, healthcare workers. They tested forest workers. By that they meant like people who worked for the Forest Service, who were out in the wilderness wandering around, living in little cabins maybe. Healthcare workers were individuals who had worked with those HPS cases in that outbreak, which makes sense, right? So a lot of viruses are spread from patients to healthcare workers if we don't know how to take precautions. So that was one area they investigated. They also looked at mammalogists, which makes a lot of sense, right? So if we have a rodent-borne illness, you have mammalogists who are out trapping rodents. You would think that they would be at risk. And I actually trapped mice before '93, and we would like eat our sandwiches in the field while we're handling mice. And you wouldn't necessarily do that kind of work now, but so they actually tested people. Mammalogists, they tested close to 1,000 mammalogists. Only 8 of them actually had antibodies for SNV, so less than 1%. And none of these other areas, none of the individuals that they tested carried SNV. So, which still leads to the idea of, well, why do some people get HPS? Another study that came out at the same time was looking at those 70 of those confirmed original cases of Hantavirus Pulmonary Syndrome in the Four Corners, and looking at where they had determined those individuals had come into contact with rodents. And so based on this, if you look at this big word pops out, paradomestic, entering rodent-infested structures. So basically the bottom line here is people don't get infected because they handle mice. They don't become infected because they work for the Forest Service. They become infected because they're coming into mouse contact with mice in and around buildings. Paradomestic is just a big fancy word that means in and around buildings or in and around human dwellings. So based on this, it was actually Rick Douglas's idea, and it's pretty brilliant. If people are coming into contact with mice inside of buildings, why don't we study mice inside of buildings? And so Rick approached the Centers for Disease Control and said, why don't we do a study of prevalence and deer mice populations in and around buildings? And so they did give Rick money to do that. And that's kind of where I ended up here, is I was finishing up my degree, and somebody I knew met Rick at a meeting, and Rick said, oh, I have this project. And so my advisor came back and said, call this guy, and maybe you want to go work there. And so I came to Montana for a couple of years to work on this project, and ended up really liking it, and luckily got to stay because a position opened up stuff. So I'm really fond of mice that live in and around buildings. So thanks, Rick. All right, so what did we do? Basically, how this study was set up is we had 3 study sites that were working cattle ranches, and this really big picture is a place called the Dana Ranch. It probably has a new name now. It's been bought for some ridiculous amount of money. It's this huge ranch that was near Cascade, Montana. The interesting thing about this site, which was pretty unique, was one of those long-term longitudinal sites was close by. So if you went out down these dirt roads way off into the distance here, there were a couple of those longitudinal, 3 of those long-term longitudinal grids. So there was a lot of data from those grids. And the paradomestic portion, we trapped in and around all of these buildings here. So it was a really nice place to work. They had a lot of buildings, a lot of mice in the buildings. This, if anyone's been out in Hale Columbia Gulch, that is a ranch that is out that way. So there were a bunch of buildings at this ranch as well. And this is out near Fairmont or Grigson. And this is what the inside of these buildings look like. They're really pretty. So people use ranch buildings for lots of different things. My friend Jamie's back there. She lives on a ranch. You guys use your buildings for lots of different stuff. So we have workshops in buildings. People calve in buildings. They store junk. They change oil. So this is the areas that we were trapping in. And you can see a little mouse here and a little trap. So basically, we use the same procedures that we did in those other studies I mentioned. We trapped for 3 nights a month, took blood samples, took demographic information on the mice. This is like one of the few data slides I'll actually show you, but one of the interesting things that we found from this study was that the infection rates in deer mice that live in and around buildings were way higher than those in more natural settings. And I'll show you a comparison in a minute here, but just looking at some of these numbers, 20%, 23, 29%, and combined between those sites about 25%. Compare this to the desert Southwest, where prevalences in deer mice populations in New Mexico are something like 7%. Okay, so we have deer mice in Montana everywhere. We have really high rates of infection with SMB in those populations. This is looking at comparing with our sites in Montana. So the Sylvan sites were those long-term longitudinal sites. The paradomestic sites are these 3 sites. And this is looking at average monthly prevalence, so the average number of samples each month that we got that were positive for SNV antibodies. And we also looked at this between that Cascade site, where the other Sylvan, more wild trapping grids were about 1 kilometer away. So we see a big difference, a lot higher prevalence in mice that live in and around buildings. And the other thing that we found with this studies was that males were more likely to have SNV antibodies than females. Adults were more likely to be infected. That was nothing new. That was similar to what we have been finding in those longitudinal studies. But if you think about it, one explanation for why the paradomestic sites could have a lot higher antibody prevalence would be because you have a lot more males in the population, right? So if more males are infected and you have a really male-biased sex ratio in those paradomestic populations, that would explain it. Also, if you had a lot more adults, older individuals in those populations. So those were other factors that we looked at as well, and what we found was that the demographics between these Sylvan populations and the paradomestic populations were really, really similar. So that wasn't a good explanation for why we see these higher prevalences. Around the same time, we actually, these are ranches, right? Not everybody lives on a ranch. What does this mean for people who live in homes? Around the same time, actually, because we would periodically get on the radio or the news, people would call us and, I have mice in my house, can you get rid of them? So what we ended up doing was giving traps to homeowners who complained about this, and then we would come and get their mice and bleed their mice. And these sample sizes aren't very big, but it was kind of unique because this study was looking at mice that were actually captured in people's homes and garages, basically. And we still find really high prevalence. So we had 20% of the mice that we tested from homes in Butte were positive for SNV antibodies, and about 28% of the homes actually as well. The other thing that we did with this data was, you always hear that mice go into buildings because it's cold outside. So it's the fall and they venture in because they get chilly and they want to go inside your nice warm home. And so we actually did look at when mice were actually trapped in buildings. And we found that they actually were trapped during all months of the year, except for January. I don't think we had any during January. The little black hatching marks are when we actually caught positive mice. So what we see is we would have positive mice in March, April, May, and then another little bout in October and November. And those actually really match what we know about SNV dynamics in both these paradomestic populations and these Sylvan populations, is that we do have a peak in prevalence when the breeding season starts, which is like right around in here, and then another one when young are dispersing from their natal areas. And that's also when people get infected as well, mostly. So the good thing about science is you always have more questions, right? So our data, actually we found some good things, but it raised a lot more questions. Mainly, why do paradomestic populations have such high rates of seroprevalence? And so to answer that, I want to kind of go back to this idea of transmission. So we started thinking more about this transmission thing. So I said people get infected because they inhale virus when they're cleaning out a barn or something in these paradomestic settings. But mice can become infected the same way. And some of the work that had been done on Pumala in Europe had shown that. So mice actually can breathe in aerosolized virus and become infected that way. So one of the things we were sort of interested in is that a good mechanism, or is that more of a mechanism that comes into play in these paradomestic settings? Obviously, that's kind of hard to get a handle on because it's really hard to measure shed virus and determine whether virus is live. But we did a couple different things to look at this. And one of the things that we were curious about is home range size and how much movement goes on in these sylvan versus paradomestic populations. And so we did some radio tracking of mice from both these types of populations, which always sounds fun, but it's actually, you're running around in the dark with a collar. I should have mentioned mice are active at night, so a lot of this, like when you're doing behavioral work, it's at night. I'm not great at taking pictures, so I got these off the internet, and this mouse looks like it's dying, doesn't it? Our mice never look that bad when we put a radio collar on it. Yeah, we cut our antennas off as well. And I don't think our collars were quite that big. He looks like he's ready to climb Everest with that big pack. But basically, that's the gist of it. You have a radio transmitter around the mouse's neck. It was fun despite running around in the dark, but one of the things that we found was that paradomestic deer mice actually had a lot smaller home range sizes than these sylvan deer mice. They also spend a lot of time in buildings, so even though they're running around at night and they're running around outside, they're actually running around a lot inside of the buildings. So about 60% of their active time were actually inside of these buildings. And so the take-home message from this study that we took home anyway was that these smaller home ranges may cause that virus that's being shed in the urine and feces to be concentrated in smaller areas. So if you're only traveling a small area, we have a buildup of virus in those areas. The other thing, and they've done some studies on this SMV is really susceptible to UV radiation, so UV radiation kills it fairly quickly. So inside of the buildings, we're going to have increased environmental persistence of those viruses. And again, as I mentioned, it's hard to get a handle on, is that really the mechanism that's playing? So one of the things that we turned to was looking at population models and epidemiological models to look at which of these modes of transmission might be more important. This is Tomas Gideon. He's a mathematician at Montana State University. And so I worked with him on developing these compartmental models, which are an epidemiological model that a lot of people use to look at transmission and different modes of transmission in terms of diseases. I'm not going to pretend that I understand that. That's from our paper that we did. So he did a lot of the modeling. I did a lot of providing the data for the population parameters and explaining why some of his ideas weren't biologically relevant. So it was kind of a fun interaction. And basically, the models that we developed predicted that direct transmission, that biting of mice, is more important for maintaining virus in those natural or sylvan settings. But both of these pathways, this indirect pathway where mice are inhaling virus versus biting, were more important in that paradomestic setting. Another way to look for a virus is to try and figure out when virus is being shed. So kind of the next step that we took was to look for actual virus in the blood. So that there were some studies that indicated that might be a predictor of when mice are actually more infectious. And so if you have higher rates of virus in the blood in these paradomestic settings, paradomestic mice might be shedding infectious virus more common than sylvan virus. To do this, you have to go into the lab, and so I ended up learning a lot of different lab techniques. I'm trained as an ecologist, so I suddenly was becoming a molecular person, which actually is fun, but I wouldn't want to do it all the time. One of the things that we did was then extract RNA from the samples, and then you're going to get some mouse RNA in there as well. So you use primers that are specific for viral RNA, and then you amplify those using reverse transcriptase PCR. I won't bore you with all the details, and if you're a molecular biologist, that might be interesting. But basically, we were targeting specific regions of the virus, and then we were able to determine if that virus was present in the blood. And we actually did that for both paradomestic and sylvan deer mice. And we actually did not find a difference between the two populations. So the percent of virus in the blood was really, really similar between those populations. So this idea that maybe paradomestic mice are infectious more of the time than deer mice really didn't play out. We also did find that the virus in the blood was higher during the breeding season, which would make sense because that's when we're seeing a lot of prevalence in the mice. It's also when we are seeing people getting ill. So the next place we kind of looked at was how do people fit into all of this? I love this cartoon. I don't know why. I love the little mouse swimming pool. All right. So what do people do when they have mice in their buildings? I'm going to pretend you're my class. You have to answer me. They trap them, right? So if you have mice in your buildings, you're going to try and trap them. And so we looked at, and that's great if your buildings are mouse-proof and mice can't get back in. We were curious what happens in these ranch buildings where mice can move in and out of the building, even if you remove them. So we actually did a study with a bunch of undergraduate students, which was really fun over the summer, at a couple different sites in Butte where we were looking at, on the outskirts of Butte, basically, where we're looking at similar sized buildings, and some of our buildings were control buildings where we just trapped and marked the mice and let them go back in the buildings. And then removal buildings where we took mice out of the buildings far, far away and released them far, far away. And really what we ended up finding was that when you trap mice in an un-mouse-proof building, all you do is suck in a lot more mice. And the chance of bringing in a mouse that is antibody positive or that is shedding SNV increases. Don't you like my scientific term, suck in? What I meant was mice immigrate in from the surrounding areas. And so you can look at the numbers there. Over the course of the summer, buildings where we were removing mice had on average about 14 mice in those buildings over that time, compared to our control buildings where we only had 6 mice that were happy and they lived there and stayed there pretty much the whole time. So basically we didn't, in some of our buildings, did not get positive mice until we started removing them. And the CDC actually used this data, which I think is cool, to change their recommendations. So their recommendations before this were to always trap mice. After we came out with this, they basically said, don't trap mice unless you can mouse-proof your building. We also did, I'm running low on time, so I'm going to go maybe skip a few of these. But basically, one of the other things we did that was fun was look at what attracts mice into buildings. So you always hear mice go into buildings when it's cold, or mice go into buildings for food, mice go into buildings for bedding. Nobody had actually really tested that, so we actually created what we called mouse hotels, which were these little experimental buildings. They only had, this door was shut when the mice were active, but there was only like one way into this building. Originally, we tracked mice activity looking at fluorescent powder and how many tracks we had in these buildings. Later, we got more sophisticated. This is a pit tag receiver and antenna. So pit tags are these little passive transponder tags you can inject into mice, and so you know who each mouse is. When they come in through the antenna, the little recorder will tell you that mouse number 12 came in at 6 o'clock, something like that. And then it'll tell you how many times mouse 12 came in, and we know who mouse 12 is, so it's kind of cool. And one of the things that we actually found out was that deer mice will go into any building that they can get into. So they don't care what's inside. but they will stay longer and hang out longer and run around more in buildings that have different treatments in them. So our treatments were food, bedding, and then we had control buildings. So obviously, what would you think? Food. Mice are going to go and spend more time into buildings with food. But if you leave your porch door open, they are still going to come into your house because they're curious. Obviously, this isn't a deer mouse, right? Has anyone seen this? I put it up there because I was like delirious when I was making my last slides. And tiny hamster eating tiny burrito. Google it. It's hilarious. And these were just cute. Apparently there's tiny hamster eating turkey dinner, but I haven't watched that one yet. We also started looking at what was going on with people and what people actually use their outbuildings for. So Bobby Klein was a biology student who went off and worked for a while and then came back to get a industrial hygiene degree. And she was really interested in what people actually use their buildings for, what ranchers use their buildings for, and whether that puts them at risk for exposure to SNV. And so she actually did a study where she surveyed ranchers to determine when they use their buildings, how often they go into them, what they use them for. And basically, obviously, she found that there was seasonal differences in what they do. So in the spring, a lot of times they use their buildings for calving, things associated with that. But really one of the most common uses of the buildings, and the one that people went in and out of their buildings most frequently, was to retrieve and put back tools. So they wouldn't go into their buildings for very long periods of time, but they went into their buildings pretty frequently. And so an offshoot of that, we had another student, Kyle Richardson, who did an interdisciplinary master's degree. And Kyle was interested in taking Bobby's work and looking at, do certain activities generate more particulate matter? So if you're inhaling SNV virus, basically it's because dust is kicked up in the air and that virus is on the dust particles. And so he actually did some air sampling in different ranch buildings. And so he was looking at what time of year you have more aerosolized particles. He was also looking at what activities inside buildings actually kick up the most dust in that breathable zone. So he was measuring that in areas where people would inhale it. And so basically what he found was that actually walking around in buildings kicked up a lot more inhalable particulates than actually sweeping. Think about it, sweeping, you're kind of kicking up stuff down close to the ground, so it wasn't really as much up into that breathable zone. Obviously, it's going to kick up some dust. So that was kind of an interesting study. Kyle is actually in New Zealand now working on a PhD doing disease work there. So I'm just going to go for a couple more minutes and tell you kind of what we've been doing recently and where we're going with all these. These are a couple of my colleagues. This is our Three Musketeer picture that we took recently. This is Angie Lewis. She's a population disease ecologist modeler. She just got a job at University of Montana, so she's there. This is Karun Begamian. Karun is doing a postdoc in Florida, but she has a lot of experience with mice. So the three of us are working on a couple different ideas and proposals. This is Karun's work. I'm not going to talk about it. I just wanted to show it to you because it's kind of cool. Karun did her PhD at Emory University in Atlanta, but she actually did her research here. And what we were interested in was looking at disease transmission in an experimental setting. So Karun calls her talk How to Run a Mouse Ranch in Montana, because basically that's what she did. She had these enclosures where she could control the number of mice that were actually in them, and she knew the number of infected. And she was looking at if you put infected mice in here, do you get transmission to occur? So she was looking at high-density mice. Do we get more transmission in high-density populations versus low? I don't have time to talk about what she found, but it was pretty interesting, and it was recently published. What was kind of interesting about this work was that while Karun was doing it, she actually developed a more sensitive protocol and extraction of RNA method and a different PCR procedure that allowed us to be better able to detect viral RNA in the blood, which is kind of cool. So this is a paper we just had. So before I mentioned that we had like 20% of our samples had SNV RNA in the blood, which we thought was infectious. With this new protocol, and we tested it on some of the old samples that we had done before, and then some new samples, we actually get much higher percentage of mice with SNV RNA in their blood, so 77 and 75. We also got a big difference between males and females as well. And then this I'm not going to bore you with, but basically the bottom line with this is, along with Corinne's work, she was able to, at the time we did our work, which is now is probably about 8 or 9 years ago, we didn't have the capability to measure the amount of virus in blood. There are machines called real-time PCR machines that will do that. They used to be really, really expensive. And in the last couple of years, we just got one that we share between chemistry and biology. So one of the things that Corinne and I are going to do is look at the amount of virus actually in blood and how different factors affect that. So what Corinne found was that when virus loads were really, really high, those were the mice that were able to transmit SNV to other mice in her enclosures. So we just submitted a proposal where we're looking at kind of this whole dynamic. We're looking at how weather affects density, how weather affects stress of mice. So we're going to measure stress hormones in mice. And then we'll be looking at how that plays into this idea. We're testing both saliva, blood, and feces for virus and quantifying the amount of virus in that, which actually will be pretty fun. The other thing, and then I'll leave you, deer mice actually in Montana, we have deer mice everywhere. They have a higher prevalence, especially in and around these buildings. But we really don't have as many human cases as we get in the desert Southwest. Rick did a paper on this where they were adjusting for urban people versus non-urban, which was actually pretty interesting and shed some light on this. But we also have a really lower fatality rate, which leads to this question: is the virus, the SNV variant that we have in Montana, different? I'm going to skip these for the sake of time, but basically, so that is where another project that we've been working on is sequencing the virus from a bunch of different sites in Montana and looking at it. I guess I can flip back one. And what we find is at the nucleotide level, we do find actually quite a bit of difference between strains that have been published in New Mexico and California. But when we look at the protein level, not as much. We have smaller changes. We were a little discouraged about that, but recently some folks at Rocky Mountain Lab, Dave Safranetz, they developed a model, a primate model for HPS infections, so they can study how HPS affects animals. And they're using macaques. I always say that wrong. But basically what they found was that the difference between the virus that they could actually infect the mice with and one that wasn't infectious, they actually had to pass it through deer mice first, and it only had 2 nucleotide changes. So this is kind of exciting, which might suggest that even though we don't have huge changes between New Mexico virus, that the small differences that we have might explain this infection thing. And so actually we just got samples we had samples from all these places, and we just got samples from Yellowstone and eastern Montana as well. So obviously, anytime you do a lot of work like this, there are a lot of people to thank. I have them all listed up here. Funding is mainly from the Centers for Disease Control and also from NIH. Lots of landowners let us do crazy things on their lands, so they all need to be thanked. Lots of great people at the CDC who have been really supportive. And then all of these folks are mouse trappers or lab workers. A lot of them are undergraduate students. And I think it's better to kind of look at it that way. So we never have fun. Don't we look like we're really bored? No, it's actually been really super fun. And with that, I will take questions, if I have time. We all need those hats. No one? Bill? Yeah, I mean, that's partly it. Males probably do fight more. You're talking about the deer mice, not humans, I'm assuming. Yeah. The other difference is the Sol virus, the virus carried by Norway rats. They've done a lot of studies looking at hormonal differences between males and females and how that affects the immune system. So what they find in those viruses, and nobody's done those studies in hantavirus, is that a lot of the male sex hormones actually suppress the immune system, so males are more likely to be infected. So there's some interesting more work that could go from there, yeah. And then males are more aggressive than females in terms of during the breeding season as well. So, and they have more wounds, yeah.
You have had mice with pet rats that are high risk of getting contracting.
Oh, it depends. If you've had mice for a long time, like you mean like little white mice in cages in your house, pet mice. Deer mice, I don't know many people who keep them as pets. The other mice don't actually carry it. And so if you have pet store rats, your case of getting like the Sol virus, yeah, is pretty much non-existent, yeah, so you're fine.
The first person to die of hantavirus and have deer mice.
Yeah, I get crazy calls every once in a while, and there was some woman who called me from back east, and she was telling me about, my husband's a retired gynecologist, and he's really bored, and now he thinks he's an animal behavioralist. So he had trapped all these deer mice and was like keeping them in their house and watching them, and she goes, I keep telling him they're dangerous. Will you call him and tell him? So I called this guy, told him he shouldn't do that.
Curiosity question. A lot of the very fascinating modeling work that seems to be happening right now is derived from Earth climate.
Mhm. El Nino. And that doesn't hold everywhere, but there was a lot of rain and a lot of vegetation and a lot of insects. And the deer mouse populations were really, really high during those years. Yeah. So So the one area that we're really interested in is looking at how climate change will change disease prevalence. And so that's, I'm really excited about Angie Bean and Missoula, because she's a really good modeler. So that's some of the questions we're asking, is looking at how climate change will affect disease. And not only here, but pretty much everywhere. That's a question we don't know.
Amy said it's a lot more complicated than what you expected. She's done it several times in models. It's what you might call positive results, but it's not really very satisfactory. It's very complicated.
Doug?
You mentioned that one of the variables you were looking at was mouse stress. Do you bring them to a mouse psychologist?
We do. Would you like to help with that? You actually, there are certain hormones, so you look at certain hormone levels, and so you can extract a lot of the stress hormones out of feces. So that's how we're going to do it, is take fecal samples from the mice, which are actually really easy to get, and then there's different ways of extracting the hormones out of that and measuring them. Yeah, we don't talk to, I'm not the mouse whisperer. I don't talk to the mouse. I do like the tiny hamster eating burritos, but I don't talk to my mice. Anyone else? Yeah, that might be, there's also a high prevalence. There's probably an easier way to do this. There is a high prevalence among natives as well. And so originally, a lot of the outbreaks were on the Navajo reservation. So you're talking about this one, I think. Yeah. No, that's all right. Right here is what you're looking at. Yeah. So if you look at the areas, actually the white thing is pretty much, if you look at the distribution of the cases, yeah, it's just the population. but they make up 20% of the cases. So, and that could be a more of a lifestyle thing. So on the Navajo reservation, a lot of these buildings that people were living in were not mouse-proof. So mice were, when populations were high, mice were moving in and out. Other questions? All right, thanks everyone for coming. So we just skip.